back, ladies and gentlemen. We are going to focus our video today on the process of mitosis. Yes, that is how cells make copies of themselves and divide. So we're going to look at how those cells actually go through the process and divide. And we're also going to take a little bit of look at how the chromosomes that are located inside of the nucleus divide and replicate to ensure that you have the same number of chromosomes in each daughter cell that is produced at the end of mitosis. A little bit about our objectives for this particular video. Hopefully by the end of this video you should be able to explain the importance of the cell cycle, detail each step within the cell cycle, and understand what results from the process of mitosis and, you guessed it, the cell cycle. We have a couple of key vocabulary terms that we are going to go through and discuss this unit. The first one being the cell cycle. We'll talk about different phases within the cell cycle, including G1, S, G2, and then we're going to go through the process of mitosis, which includes the phases of prophase, metaphase, anaphase, telophase, and then of course the physical division of the cell known as cytokinesis. We'll also discuss concepts related to the metaphase plate and interphase. So the cell cycle is very important as cells grow and divide to sustain life. As you know, we all start off as a single-celled zygote and then undergo mitosis to become the multicellular organism that we are today. Well, this is a very distinct set of steps that we go through in order to perform cell division. So if we take a look here at the image on the right hand side, we can see that most of the time that is spent in the cell cycle we are spending in interphase, which is simply just the growth and the DNA replication portion of the cell cycle. When I talk specifically about mitosis, I'm talking about the division of the chromosomes that are located inside of the cells after they have been replicated. And the last step, of course, is cytokinesis. If you actually break that word down, the word cyto means cell, and kinesis, like the word kinetic, means moving. So when I talk about cytokinesis, we're actually talking about the physical division of the cell. Now, we can break each of these down into smaller steps, but keep in mind that the cell cycle is primarily divided into three major steps: interphase, mitosis, and of course cytokinesis. So if we take a look at interphase a little bit more deeply, we look at the phases of the cell cycle, and we have the gap 1 phase, which is known as G1, and that's just normal cellular function. Most of the time, your cells are going to be spending time in G1. It undergoes S phase, and that's the phase where DNA replication occurs. If you remember from our molecular genetics unit, we talked a lot about the process of DNA replication and how chromosomes make physical copies of themselves. The step after DNA replication is called gap 2 or the G2 phase, and that's just continued growth and preparation for mitosis. So interphase is the primary phase in which the cell actually lives its life. We have a phase for normal cellular function, a phase where the DNA makes a copy of itself. So again, if you remember, we go from single chromosomes to the chromosomes that look like X's during the process of S phase. And then gap 2 is just continued growth of the cell. Now, the next steps are the actual physical division of the chromosomes, and this occurs during the phase known as mitosis. We can break mitosis down into 4 individual phases: prophase, metaphase, anaphase, and telophase. So during the first step of prophase, the chromosomes actually condense. So chromosomes exist kind of as bunches of genetic material. Well, they condense into the chromosomes that you and I think of when we think about the large X's that appear inside of the cell. Spindle fiber appears from the centrioles. If you remember way back in our cell unit, we talked about how centrioles actually aid in cell division. Well, you'll see how that takes place coming up here soon. But spindle fibers are like little tiny protein fibers that appear from the centrioles, and they're going to be used to pull apart the chromosomes in later phases. Furthermore, the nucleus actually disappears, so the chromosomes are actually starting to roam freely in the cytoplasm. So that's essentially prophase. Now during metaphase, the spindle fibers actually attach to the centromeres on the chromosomes. They attach to the central parts of the chromosomes and push and pull them so that they're located in the center part of the cell. They line up on what is known as the metaphase plate, which is again that center part of the cell. So what you have now is you have your centrioles in opposite poles of the cell, and all of your chromosomes are nice and neatly lined up in the center of the cell, which makes them ready for the next step. The third step is known as anaphase, and these chromosomes are actually pulled apart. So we go from those X's, each half of that replicated chromosome is going to be pulled apart. Those spindle fibers contract, which pull the chromosomes apart, and they're moved towards opposite poles of the cell. During telophase, the chromosomes decondense, the nuclei reform, and the spindle fibers disappear. So what's really nice about this is that telophase is almost the exact opposite of prophase. Remember, in prophase, our chromosomes condense, our spindle fiber appears, and our nuclei disappear. Well, in telophase, the chromosomes decondense, so they're becoming more along the lines of normal DNA or normal chromosomes. The nuclei reform, and the spindle fiber disappears. Now, the only step left is cytokinesis, when the cell actually physically pulls apart, leaving you with two cells that are genetically identical to one another. So really think about what we get at the end here. So even before replication, we have a cell that has 46 chromosomes inside of it. Those cells undergo the DNA inside of the cell undergoes replication, and then the process of mitosis occurs, which pulls each half of the replicated chromosomes apart. Thus, at the end of mitosis, we end up with two cells. Those two cells are genetically identical to one another because the DNA underwent DNA replication during the S phase of interphase. And because they're genetically identical, they have 46 chromosomes in each new cell, which is the exact same number of chromosomes that we started with prior to the chromosomes replicating in S phase. So, long story short, at the end of mitosis, we have two cells. They're genetically identical to one another, and each cell contains 46 chromosomes. So keep in mind that the cell cycle is made up of three major components: interphase, mitosis, and cytokinesis. Mitosis is made up of four main stages: prophase, metaphase, anaphase, and telophase. And then at the end of the cell cycle, we have two cells which are genetically identical to one another and contain 46 chromosomes each. Hopefully you understand the objectives for this video. Hopefully you can explain the importance of the cell cycle, detail each step, and understand what results from the process of mitosis in the cell cycle. Again, we'll talk a little bit more about the process of meiosis and how it's very similar to the process of mitosis in our next video. Have a great day, guys. We'll talk to you later. Bye-bye.